Hi guys, this is the last video in a series of videos I'm doing on hepatitis viruses. I do kind of an overview of what a hepatitis virus is and an explanation of prodrome, preicteric, and icteric phases in the hepatitis A and hepatitis E video. So if you want to know more about that, check out that video. In this video, I'm actually going to talk about hepatitis C virus, or HCV. HCV is kind of an interesting virus to me. When I was kind of coming up in school and grad school, being an immunologist and microbiologist, HCV kind of served as a perfect storm of a chronic viral infection. And then somewhere around my last year in grad school, they actually figured out ways to treat and potentially cure chronically infected HCV patients. So it's really exciting anytime we've got some illness that was a big problem for years and years that now we're able to actually treat effectively. So it's a very exciting virus to me to talk about because it's changed so much just in my lifetime. But let's just talk about HCV, the virus. So first off, one of the things I always mention is that typically when I talk about hepatitis viruses, I think people think about them the same way they think of like adenoviruses or rhinoviruses, that, it's all in the same family. And that's true for rhinoviruses, but hepatitis viruses are referred to as hepatitis viruses because they affect the liver, not because they are their own family. So HCV is actually a flavivirus. That's the viral family it is in. And therefore, it has a plus sense RNA genome. It can be spread in blood or needles through sexual contact and perinatally in utero. It has kind of a median incubation time of 14 to 180 days, so highly variable. And it has kind of, one of the reasons we have this super variable incubation period is that it has kind of this insidious start to icteric symptoms. So if you think about the three phases of hepatitis, you have prodrome, which is pretty much like any other infection. It's characterized by fever and myalgia, maybe some, discomfort, but nothing very specific. Then we go to the preicteric, and in the preicteric, we start to actually see some of that discomfort as a result of the liver. So you might have some right upper quadrant pain, anorexia, food aversion, nausea, maybe some vomiting. And then by the icteric, that's when we see, jaundice, clay-colored stool, things of that nature. So with HCV, we see kind of this insidious slow start. The other thing to note about hepatitis is that it has a high potential to go chronic. It becomes chronic in about 70% of patients, and it's a huge cause of hepatocellular carcinoma or cancer. And it's very, very prevalent throughout the world. So those are kind of the main things I think about when I think about HCV. Okay, so we are going to go through now what kind of makes HCV the virus it is and why its replication and kind of the dance it has with the immune response is so intrinsic to how it establishes chronic infection. So as I already mentioned, HCV is a flavivirus, but it's not a flavivirus that is spread by mosquitoes like many of the others. This one is spread by blood and perinatal infection, things like that. There are 6 major genotypes, or sometimes referred to as clades. And between or within each genotype, there's a lot of genetic diversity, which means there's a lot of antigenic diversity, which further hampers the immune response and its ability to identify antigens and fight them. It has a positive sense RNA genome, and it is enveloped, and there are numerous proteins involved. Okay, so there's 2 glycoproteins, and the glycoproteins are E1 and E2. The rest of the proteins tend to be these non-structural proteins, okay? And these non-structural proteins kind of makes them seem like they're not important, but they're actually really important because they have enzymatic activity. And that's really important for efficient viral RNA replication. So NS3, NS4A, NS4B, and NS5B. And actually, a lot of the new anti-HCV drugs that we have to treat this infection are actually targeting these NS proteins. There's also a viral RNA-dependent RNA polymerase which is really error prone, which helps create that genetic diversity that then therefore becomes a part of antigenic diversity. And that also makes vaccine design very difficult. It's us and the chimps that tend to get this one. Another way we are close to that ancestor. The receptor for it to getting into cells is CD81, which is also known as tetraspanin. There's also a scavenger receptor, and tight junction proteins, claudin and occludin, are all ways for this virus to get into host cells. Okay, so let's kind of talk about the replication and then the immunopathogenesis, even though that seems a little bit backward based on what I have on the slide. So I'm not going to go through the full replication cycle like I have for HBV or HIV. And part of the reason for this is that really we're only targeting kind of one spot, and that's those non-specific or non-structural proteins. HCV does not grow well in culture, so it's somewhat hard to study, which kind of hampered our treatment efforts for a long time. There are 7 genotypes. Only 2 genotypes have recently been cultured successfully. And the steps described here are from the HCV genotype 2 system and from flavivirus replication. So we're kind of putting it together. It's hard to study the replication of a virus that you can't grow. And the envelope proteins might be associated with serum lipoproteins for attachment. The E2 protein may bind CD81, but some of this is still really up in the air. So basically, things you would want to know about HCV replication. One, the replication takes place largely in the cytoplasm, okay? Which makes sense. Remember, it has its own viral RNA-dependent polymerase. Its genome is translated first after uncoding to make a polyprotein. The polyprotein is then cleaved into individual structural and non-structural segments or proteins. The genome does encode for its own RNA-dependent RNA polymerase, and that's used for transcription of the RNA, which makes sense because that's what those do, right? Okay, and then the non-structural proteins actually form a little structure with vesicle membranes to make a complex to support RNA synthesis. And this RDRP is really error prone, and that increases potential for mutations and variations to be generated. These non-structural proteins are the target of many of the anti-HCV drugs, so just something to keep in mind. Okay, so let's talk about the pathogenesis and immunity. One of the things that I find really interesting about kind of all of the hepatitis viruses is how important the immune response to the virus is in kind of the disease that we see in patients, okay? So HCV can remain cell associated and just prevent host death. It can just hang out in the cell, and that's one of the ways that it can actually go chronic, is that once it's in there, it can just kind of hang out for the lifetime of that cell. So this is just some virions hanging out, no problems here, nothing to see here. And in that case, you're not necessarily going to see any symptoms for a long time until you actually see damage. The virus is able to inhibit apoptosis, and it also, I don't know if I'm showing it well here. Hang on, let me find it. Okay, so the virus itself, if this is my virus, it's actually able to inhibit interferon, specifically type 1 interferon production. So when you inhibit the expression of interferon alpha and interferon beta, you actually inhibit kind of the cell's immediate surveillance system to tell all of the other cells around it that there's been an invasion, right? So interferons essentially shut down protein synthesis and increase MHC class I expression in nearby cells. It's kind of the save yourselves cytokine. And when they shut it down in a singular infected hepatocyte, then it actually inhibits the ability of nearby hepatocytes to protect themselves. And it actually does this by binding to the TNF receptor and to protein kinase R. So those are two kind of proteins that are involved in this interferon synthesis process. These actions basically prevent the death of the host cell and promote escape from host protections to promote persistent infection in more and more cells. The cell-mediated immune response is really, really key for this. And that's why a lot of times what we see is that patients have been chronically infected with HCV for years, and then as their cell-mediated immunity wanes, as they get older, or some sort of major life event happens, like a cancer diagnosis, a transplant, an HIV infection, then we actually start to see more and more of the damage. The cell-mediated immune response is important to kind of resolve the infection. So if you're talking acute HCV, you're going to see a really strong CD8 immune response. And the CD8 immune response, and I'm just going to kind of map it out here, is what's actually going to recognize the viral protein expressed on MHC1 from the hepatocyte. And there it's just going to do what CD8 cells do. It's going to produce interferon gamma, TNF alpha. It's going to degranulate, leading to release of perforin and granzymes. It can also perform FASL mediated killing if the organism is expressing, or if the cell is expressing the ligand. Of course, in order to have CD8s, we also need to have help from CD4 positive T cells, because the CD4 positive T cells produce the interferon gamma, IL-2, and IL-12 that promotes the CD8 T cell response. That also makes for better antigen presenting cells, right? And also recruits, I don't know if they're here, but NK cells. Now, the other thing I'll say is that anytime you have CD4s and you have B cells, you have a nice symbiotic relationship, right? Because the B cell can act as an antigen presenting cell, and the T cell then gets stimulated by the antigen presentation and provides cytokines. The cytokines will tell the B cell to secrete antibodies, but here's the kicker. Antibody isn't really protective, because remember, we either fight this thing when it's acute with a really strong CD8 T cell response and NK response, which is going to lead to hepatocyte damage, or it establishes chronic infection and the CD8 T cells become exhausted by expression of CTLA4 and PD-1, right? So those are kind of our options. Fight it early or live with it forever and your CD8s won't work anymore. Once we're living with it forever, the virus is pretty much in the cells. So our antibodies aren't really going to help. They don't seem to really neutralize well. The one thing that they might help with is some ADCC using NK cells early in acute. But again, the antibody response to HCV is not protective, okay? Okay, so let's talk about all of the things from clinical syndrome through diagnosis and treatment at this point, okay? HCV is one of the hepatitis viruses that can lead to acute or chronic symptoms, okay? So anytime we think of a hepatitis virus, we have to think of our three phases, right? We have prodrome, which is kind of non-specific viral symptoms. We have pre-icteric, and then we have icteric, okay? And it's really between this pre-icteric and icteric when the liver itself is actually showing signs of damage. 70% of patients who contract HCV actually wind up developing chronic hepatitis. It's very widely prevalent. There's about 170 million chronically infected patients. And part of the reason we call them carriers is that they might not know that they are infected until they hit this icteric phase, which sometimes doesn't happen until much later in life. How are you going to diagnose this? Well, lab evaluation, if we're looking at acute hepatitis, you pretty much always start looking for AST and ALT, okay? And your practitioner and pathophysiology discipline directors can tell you more about that. But anytime you see those elevated ALT or AST, usually ranging from 4 to 8 weeks after exposure, this is going to take a little bit of time to actually lead to this result. That kind of makes me think acute hepatitis. The peak of transaminitis is usually during the time of jaundice, so during that icteric phase. And jaundice is usually evident once bilirubin levels reach about 2.5 milligrams per deciliter. Diagnosis is made typically by ELISA using anti-HCV antibody. So you make antibody, but remember, it's not really all that protective. You can also do detection of the RNA genome, and that's actually the gold standard for diagnosis. But oftentimes you'll see both tests run. And the reason for that is also that detection of the genome tells you later how well your therapy is working, because once you have the antibody, you're pretty much always going to have the antibody. Seroconversion or detection of the antibody takes a long time, 7 to 31 or 30 weeks after infection. And so it's not always detectable in viremic people, particularly if the patient is immunocompromised. So if you have an HIV positive patient with a really low CD4 count, they might not be able to make antibody because they might not have enough CD4 help to basically inspire the plasma cells to differentiate and make antibody. There is such a thing as an indeterminate antibody test. Basically, it's a negative result in a high-risk patient with icteric symptoms or a patient who is immunocompromised or on hemodialysis. And in that case, those patients should always just be tested with RNA-PCR because, like I said, that's the gold standard. And then once you put the patient on treatment, you can actually track their RNA levels to make sure that your treatment is working and make sure that it's actually decreasing. If the RNA levels are remaining the same, it could indicate that if you haven't been treating them, that it's gone chronic and they have lost that acute battle and they should begin therapy, or that you need to maybe change their therapy, okay? So like I said, you can have acute or chronic hepatitis that moves into that icteric phase. Sometimes we do see cirrhosis, and this one is highly associated with hepatocellular carcinoma. So how do we treat it, prevent it, etc.? You want to try to avoid it, first off. Precautions include avoidance of bloodborne pathogens. The blood and organ supply in the US are screened for HCV, and persons with HCV shouldn't necessarily share personal care items like medical supplies, toothbrushes, etc. And they definitely should practice safe sex because that is one method of getting it. Also, because it does affect your liver, you're going to want to watch alcohol drinking. It should be limited because it actually exacerbates the liver damage caused by HCV. Chronic hepatitis, but not typically acute hepatitis B and C, can actually be treated with antivirals. So basically, we let you try to fight it in the acute phase. If you fail and you go chronic, then we might be able to treat you. It's a little backward, but it is actually how it works. You can actually review these in the pharmacology section of this self-study, where we go through all of the different antivirals for HBV and HCV. And these medications have really revolutionized what we know about HCV and how we actually interact with it. So very exciting there.